All right, so here we are at chapter 13, talking about how populations evolve. So we've gotten past the mechanisms of heredity, but what happens on the population level? So after seeing how, you have parents and the offspring that you can predict would be derived from those parents, what happens on the larger scale that determines how groups of a single type of organism change? So this unit in the book is referred to as evolution and diversity. Now, back in 1955, the World Health Organization set out to eradicate malaria. And they figured the best way of doing this was to kill the vector, the mosquitoes that carry the parasite from person to person. And DDT was a recently developed pesticide, and so they took that and they went spraying a huge area of land, trying to basically wipe out the mosquitoes, and with it, the ability for malaria to be transmitted to humans. Now, it did immediately kill most mosquitoes, but there were survivors, right? There were mutations in some individuals in that whole population that conferred resistance to DDT, allowed them to survive DDT, so that handful of survivors gave rise to new DDT-resistant populations. Right? This would be actually observing evolution in action. So again, things like the drug-resistant infections we talk about now, same principle. So unfortunately, eradicating malaria is no longer viewed as imminent. Right? The bombing with pesticides was not a solution on a whole bunch of levels. So by using a combination of mosquito control strategies and also just giving people protection from mosquitoes at night, like netting, they're making progress in the battle against malaria, but it's still an ongoing issue. WHO also monitors the evolution of drug resistance in that parasite's populations around the world. So this is an ongoing problem. Not so much a problem here in North America, but in other parts of the world it is really a scourge. Okay, so for all of human history, we've been naming, describing, and classifying the inhabitants of the natural world, right? People like to categorize things. They like to put things into little boxes and sort them out. Well, there's a whole science that's concerned with identifying, naming, and classifying species, and this is taxonomy. Taxon, that word literally refers to name. So the people who try and figure out what groups organisms together, what separates groups from each other. And we still use the Linnaean system. This guy, Carl Linn, he Latinized his name to be Carolus Linnaeus. The Linnaean system includes a method of naming species and hierarchical classification of species into broader groups of organisms, right? So he started from the small end saying, okay, we're going to name this organism this organism that. Oh, now we have, these guys grouped together, and these are part of a larger group, and so worked upward. He introduced a system for grouping species into a hierarchy of categories, and used a naming system with two words, binomial, right? Bi for two, nomial for name. And so every species that we've described to science has its own two-part Latinized name, or binomial. The first part is the genus, plural genera, and that's a group of closely related species. The second part of a binomial is used to distinguish species within a genus. In a very rough fashion, it's almost like if you think of someone's last name, right? It's a family name, so a whole bunch of members of the family are included in that. So our family names would be kind of like the genus, and each individual, their first name would be the species name. So we'll be using this not so much in this course, but you'll see the terms come up. So again, genus is the group of close related species. So you'd have the generic epithet, and then the second part is the specific epithet, or the species name. So again, this is one way we help understand the world. It also gives scientists a way to discuss organisms with each other. If people just used things like common names, you would have a lot of disagreement and misunderstandings. Now, the criteria used to define more inclusive groups, such as families, orders, and classes, is really ultimately arbitrary. and it does change over time. Because if you think about it, back in the day, it was just simply morphology, how something was put together. But now we can go all the way down to studying the DNA of organisms and comparing genes or proteins produced from those genes. So as the technology changes, so does our perspective on classification. Now, they give you a good example here. Which pair of animals is more closely related to each other? The first pair is Ursus americanus and Ursus maritimus, or Ursus americanus and Bufo americanus. Now, again, we tend to think, we might think in terms of our, our names first and say, oh, they share the same last name, so it would be the second pair. No. The first pair shares the genus. So those two bears are members of the same genus, whereas below is a species of bear and a species of toad. So it's the first name, which is what's shared. And so you break down that genus into all the species. Now, here's the infamous series of boxes here. And they go from bottom to top. As I put it in the review notes, I went from top to bottom. But it works either way. So let's start down up here at the top at Pantheropartus. All right, so that is one individual species of a large cat. It is one of a genus of cats that contains multiple species, right? You also have tigers and leopards and all those guys. All right, so the individual is Pardus. The species is Pantheropartus, right? So Panthera is one member of genus. Panthera is a genus containing multiple species. The family Felidae contains multiple genera, right? So there are multiple types of felines, not just the big cats. The order Carnivora refers to all the mammal carnivores, right? So you have cats, canids, cats, dogs, bears, weasels, right? There's all these major groups of carnivores. So Felidae is just one family within the order Carnivora. And Carnivora is just one order in the class Mammalia. So you have Carnivora, and then you have Rodentia and other groups of, that are orders that are included in class Mammalia. Class Mammalia is one of many one of many classes within the phylum Chordata. Phylum Chordata is obviously the one we belong to, and I'll talk about that in a little more detail when we get into later in this chapter and when we get into the diversity chapters. So phylum Chordata is just one phylum within kingdom Animalia. So Animalia would include not just things that have a dorsal hollow nerve cord and stuff like that, but things like insects, things like jellyfish, right? So all the other types of animals that are out there. And kingdom Animalia is just one of the 4 eukaryotic kingdoms, right? So if you look at the bottom picture there, right, there are 3 domains: bacteria, eukarya, and archaea. Animals, plants, and fungi, and protists, which are kind of a weird group, those are all kingdoms within the domain eukarya. So once upon a time, we just learned life in terms of a few kingdoms, but now we know it's gotten a little more complicated than that. Now, explaining the origin of this diversity of life, right? So we have this way of naming things and grouping things, but where did all these different forms come from? Well, the explanation of the origin of the diversity of life accepted by present-day biologists is the evolutionary theory, proposed by Charles Darwin in his well-known book On the Origin of Species by Means of Natural Selection, sometimes just referred to as On the Origin of Species or The Origin of Species. But he published this in 1859. So if you think about that, that's not really that long ago. This is relatively recent in terms of human history. If we go back further, we can see some of the earlier ways of looking at classification. If you go back to Grecian times, the Greek philosopher Aristotle generally held the view that species are fixed. In other words, they only have one form. It's a permanent thing. They don't change over time. All right, so as life appeared on the earth, it has always looked like that. Now, Judean Christian culture took this and reinforced it with a literal interpretation of the biblical book of Genesis. which tells the story of each form of life being individually created in its present day form. And that was held for quite a while. So if you go to the 1600s, religious scholars estimated the Earth as about 6,000 years. So this whole idea that living species came into being relatively recently and are changing in form really dominated the intellectual climate of the Western world for centuries. Now, what happened is once they started getting around the 1800s, they started really coming into contact with these things called fossils. Now, fossils are imprints or remains of organisms that have lived in the past. So when people found things like these guys, the one on the left we now know as the shell of a mollusk, a type, basically a relative of snails. But at the time, they didn't know of anything like this, so they said, "Oh, this is a snake stone. This is a snake that has died and been preserved somehow." And the guy on the right, of course, didn't really resemble a whole lot of things, so that was just a mystery. Ichthyosaurs most closely resembled things like dolphins, but it was you could easily tell that they weren't quite the same. So naturalists who were finding these fossils were comparing them with living species, right? And looking at patterns of things they had in common, things that they didn't have in common. In the early 1800s, French naturalist Jean-Baptiste de Lamarck suggested that life evolves, that life changes. And he explained evolution as the refinement of traits that equip organisms to perform successfully in their environments. Now, that part we're like, okay, yeah, that sounds about right. But his mechanism was off. He proposed that by using or not using body parts, an individual would develop certain traits to pass on to the offspring. And the classic example is that of a giraffe. All right, so a giraffe is reaching for leaves in a tree and it's stretching and stretching and stretching. And that made the neck a tiny bit longer. And that state was inherited by the offspring, which then stretched and stretched and stretched. And so it was using or not using body parts that developed traits that got passed on to offspring. Now we know that isn't the case, but at least he started the ball down the field with the whole idea of life changing. Now, on to Charles Darwin. He was born over 200 years ago on the same exact day as Abraham Lincoln, which I always find fascinating. Now, he was great with nature as a boy. He, like a lot of kids, he was out there looking at things and catching things and having a good time with that. As he got older, basically he was told, okay, now you gotta get a career, you gotta get a job somewhere. So he tried different things. He went to medical school, but he found medicine boring and surgery, because these were the days before anesthesia, was horrifying because people basically had to be held down while they're being cut open. He didn't find that good to watch. So he then, as many in his family did, went to study to be a clergyman at Cambridge University. And he wasn't sure that was really working out either. So he took a job at the age of 22 on the HMS Beagle, Her Majesty's ship Beagle. Essentially, the captain wanted polite company, educated company, so he didn't have to hang around with all those sailors and rough people that worked on the ship. And the good thing is, because that guy was a bit of an elitist twit, he hired on this guy to say, "Oh, I'll have this young man keep me company." Well, he didn't just keep the man company, but when they set to land, he was allowed to basically, go ashore and do what he wished. So this survey ship, it was a ship that was basically bouncing along the coast of South America and also a couple of other points in the world, and it's basically trying to sort out the shoreline to help map makers generate a more accurate map of South America. So when they were at port, he would spend his time on shore exploring all the living things there, just as he did as a kid. He collected thousands of specimens of fossils and living plants and animals. I always wondered what the below decks looked like on that ship. Anyway, he also kept detailed journals of what he observed, and in those journals, he carefully noted the characteristics of plants and animals that made them well suited to diverse environments. Because again, a lot of people didn't travel that far from where they were born. Now here's somebody who had a thorough knowledge of the local living things where he grew up, and now is looking at organisms in other parts of the world and realized that some things may have some similarities to, organisms he knew of, but are a little different here or a little modified there. So he realized that certain features made these organisms better suited, right? Better able to live in diverse environments. And this is just sort of a couple of illustrations. On the left, you see Darwin as he looked in 1840. Typically, people see Darwin as this elderly, bearded, bald guy. That's what he looked like as a younger man. So it's kind of nice to represent his younger years. On the upper right is a illustration of the HMS Beagle, and in between is the map of his travels. So he started out in Great Britain, and if you followed the little arrows, right, they then traveled all along the coast of South America. Now, if you are not familiar with South America geography, you've got all sorts of environments there. You've got grasslands, you've got mountains, you've got all sorts of different places. So he was able to see quite a wide range of habitats. So they bounced along the South America, rounded the tip at Cape Horn, and then made their way up. And just as they headed away from South America, in that inset you see a very special place they stopped, and that's the Galapagos Islands. So we're going to look at this a little more closely. So here's the infamous Galapagos Islands, and you can see there's a bunch of islands, different sizes, and on these different islands there were slightly different habitats. So some were grassier, some were woodsier. There was actually quite a range of environments, and the distance from each other, because if you look at that line, down at the bottom, right, 40 miles is a big distance. So it's further, if you look at some of these islands are so far apart that things could not easily move from one to the other, right? Birds were not inclined to fly, 40, 50 miles to get to another island. Certain animals like tortoises were not, really adapted to, go on long swimming trips. So generally, the things that had landed and evolved on those islands stayed on those islands, right? So you isolate them. When you isolate organisms, then they tend to change over time to suit that particular environment. And so the islands being different, the animals in the different island evolved in different ways. So many of his observations indicated that geographic proximity, living close to each other, is a better predictor of relationships among organisms than similarity of environment. So, give you a good example, the things that he knew, the things he observed in temperate regions of South America more resemble species living in tropical regions of that same continent than species that lived in similar climate back in Europe. Also, the South American fossils he found, though clearly examples of species that were no longer around today, but they definitely looked more like the modern living organisms on that continent, more than, say, necessarily just any other, old fossil of an organism. So you realize that there was this geographic proximity was an element. And he really was intrigued by what happened in the Galapagos Islands, because most of the animals that are found on these islands are found nowhere else in the world. Literally, the Galapagos tortoises are found on specific islands in the Galapagos Islands and nowhere else in the planet. So these have evolved in these places and obviously never dispersed to other parts of the world. But the thing he noticed is that they did still resemble South American species. A good example being the iguanas. He noticed that the marine iguanas had these vertically flattened tails that basically served as paddles. They helped in swimming. And that's similar in some respects, but very different from the tree-living iguanas found in the land, and the land-dwelling iguanas found on the islands, and the tree-dwelling iguanas found back on the mainland. So it's like you could see there are similarities, but obviously they're not quite the same. And one neat thing is each island had its own distinct type of giant tortoise. So we'll look at two forms here. So you look at the guy on the left. These are guys that grazed on grass like that fellow's about to do, right? So if you look at the shell, it has basically a nice little arch, and it doesn't have a very high curve at where the neck is. Well, that's because this guy spends his time eating downward. But you look at the guy on the right, and we refer to that as a saddleback shell. If you look at the shell in the front, where the neck and front legs are, you realize that it curves upward. Well, these guys feed from, shrubs and trees. So these guys have to be able to reach upward. So even though they shared common ancestors, obviously separated on islands with very different plants living on them, they evolved very different forms. So one thing he took along on the trip was a book. It was just came out before he left, called The Principles of Geology by geologist Charles Lyell. And again, it was thought that the Earth was 6,000 years old, right? That the Earth was fairly new and unchanged. This guy presented the case for a much older Earth that changed gradually over millions of years by geological processes that we have today, right? Volcanoes, earthquakes, things like that. By the time he got back to Great Britain, between reading this book and all his observations, he began to really doubt that Earth and everything living on it was specially created just a few thousand years earlier. So once he got back to England, he reflected on his observations, right? He mused about, what it was he saw in Winter's journals and looked at his specimens, talked to other scientists back in England, and he came down to the conclusion that the evidence of what he saw with his own eyes around the world was better explained by a hypothesis that present-day species are the descendants of ancient ancestors, that they still resemble in some ways. So over time, differences, from ancestor to descendant, differences gradually accumulated by a process he called descent with modification. And that was his phrase to describe evolution, that the descendants of organisms change over time. And again, this was a fairly radical idea at the time. Sort of a funny side story. He when I say he mused and read his books and talked to people, he did this for years and years, and was not really piling it up into this book we're all familiar with. But what happened was a guy who was studying beetles out in the Pacific islands in the Pacific Ocean wrote to him. This guy's name is Alfred Wallace. And he said, "Hey, I've been studying these beetles, and I got this idea that, life might be like changing and adapting to things." And all of a sudden, Charles realized that someone else was on the same trail. Now, no one stole from the other, but they both, in parallel, were coming up with the same ideas. So he got on the fast track and got that book out. I think it was within a year or two, after spending years and years and years not really publishing, anything big. He's like, okay, time to get the book out, because the first person who publishes generally gets credit for a discovery. So it was kind of funny that if it weren't for Wallace, he probably would have gone to his grave still thinking about instead of writing anything. All right, so first is, okay, life changes, but he also came up with a scientific mechanism for how it changed. And this is a process he called natural selection. Now, with natural selection, individuals with certain inherited traits, right, traits that they received from earlier generations, are more likely to survive and reproduce than are individuals with different traits that they inherited from their ancestors. And he hypothesized that as the descendants of remote ancestors spread into various habitats over millions and millions of years, natural selection resulted in diverse modifications, or evolutionary adaptations, that fit them to live in specific other parts of the world. So in other words, the marine iguana eating algae versus the land iguanas that ate terrestrial plants versus the marine iguanas on the mainland that ate leaves and trees, right? As they spread into different places and lived in those different places, they changed, right, to be better adapted to these different environments. So, I did have it in here. I'm sorry. I forgot the slide. So he spent about 20 years compiling and writing and all this. And then it was Alfred Wallace, who's a British naturalist doing fieldwork in Indonesia. he had come up with a hypothesis almost identical to Darwin's. What they don't say here is he wrote Darwin a letter basically saying, hey, can I bounce this off you? And that's what got him off his keister and off to writing that book. So he finally cranked out The Origin. And again, this supported his hypothesis with A, logic, and B, evidence, right? It's great to come up with a possible answer to a question, but if you can back that up with evidence, now you got something going. So he had hundreds of pages of evidence drawn from observations and experiments in biology, in geology, and in paleontology. So he wasn't just waving his hands. He had, as they say, brought the receipts to prove this. Now, the hypothesis set forth in The Origin has since generated predictions that have been tested and verified by more than 150 years of research. Right? If you come up with something and other people do variations on or versions of your research but applying to different problems and all that, eventually if there's flaws, they will start turning up. But this has been backed up for 150 years so far, so pretty solid at this stage. And that's why scientists regard Darwin's concept of evolution by means of natural selection as a theory. Right? This is more widely accepted than just a hypothesis, right? So you have a hypothesis, you got data that maybe supports it, maybe doesn't. If you have a hypothesis that has piled up lots and lots and lots of data supporting it, we then come up with something like a theory. So instead of just why do turtle shells look different on different islands, you can apply this theory to birds, to plants, to fungi, to all these other organisms. The same sort of patterns, the same sort of changes were observed, that things changed over time in different habitats. So now we refer to it as a theory. So it's a little bit broader in scope than a hypothesis, right? You're not answering one question, you're answering a whole bunch of similar questions. Also, it spins off new hypotheses, right? There's hypotheses about leaf shape, or hypotheses about mimicry, why some organisms imitate other organisms. So there's all these other hypotheses that spin off of this, and again supported by a large body of evidence. The more evidence you have, the more credibility your idea has. So in conversation, you always hear people say, well, I have a theory. What they're really saying is, I have a hypothesis. So if you want to be that person in the conversation, you could be the one to say, well, actually, you have a hypothesis as opposed to a theory. And you could get kicked out of parties too. All right, so evidence from fossils. Evolution leaves observable signs, right? If there were no remnants found in the modern day, how would you know how things appeared years ago? Well, we have these fossils. Now, we tend to think of fossils as the remains of an organism. It could also be things like imprints of organisms that lived in the past. It could be other evidence of them being there, and we'll get to a couple of examples in the next couple of slides. But these fossils, these imprints or remains of organisms that lived a long time ago, let you document differences between the past and present organisms, right? If you're looking at lizards, you can look at lizards from a whole long time ago and look at lizards today and see, these are patterns of differences in how the hip bones are developed, things like that. So you can make observations of the previous, long ago generations and the modern forms rather, and also realize that many species have become extinct. If you counted all the species from the beginning of life on up to today, most of the different species have become extinct. So we have a lot of diversity today, but there's so much that has already come and gone from the past. Now, the tricky thing about fossils is the soft stuff usually decays rapidly, right? You've got bacteria and fungi and things living on eating dead stuff. So that usually goes away, but hard parts, they're rich in minerals like bones or shells, may remain as fossils, right? They don't get digested and eaten up by something, and they can stick around. And say, look at this tree here. This is a fossil of a tree. Now, you could see sort of the patterns of bark and stuff like that, but keep in mind that this tree was probably buried in a wet place with low oxygen, so it didn't decay but over time, the water that seeped into it and soaked into it left minerals behind. So over very, very long periods of time, minerals replace what was the original organic content of the tree, but in such a way that you wind up with this sort of great replica just made out of stone. Now, another thing that you see are things trapped in amber. Amber is tree sap. If you've ever touched like a pine tree and it has that sticky stuff on it, what happens is tree sap can leak out of a tree from a wound or something, and something climbs on it and gets stuck in it. The amber may eventually dry out, and the sap may eventually dry out and form amber, and you wind up with this really great sample of an organism that got embedded in the amber, never decayed or anything, just preserved there for thousands or millions of years. And so sometimes we have things like this. We tend to see pictures of insects, but there have actually been lizards. I think there's even a bat down in, I think it was down at Princeton. So sometimes big things got gummed up in the amber and were preserved that way, but most of the stuff we tend to talk about are arthropods. Now, one thing that obviously was a hint that there were things that were around in the past that weren't around today, right? That's a leg bone that man is examining that was fossilized, right? It probably was embedded in silt, and minerals replaced the bony parts to leave this great replica, if you will. And there was nothing around that had a bone quite like that. So it was seeing things like that that helped lend evidence to the whole idea of evolution. Now, I said not everything is actually an organism itself. So things like footprints, right? We have Dinosaur National Park here in Connecticut. And these are footprints that were left by a dinosaur. So walking through something like silty, think of walking on the beach at low tide, right? And your footprints sink in the sand. These are footprints that were left by a dinosaur. So here you can get ideas of things like, what were the feet like? What was its gait? How fast did it walk? If you go to the Peabody, outside the Peabody, you don't have to go inside to see it. They have this great little park, including dinosaur footprints that are made, embedded into the concrete, and they have some neat things, neat stories around that. And sometimes we get real lucky and find relatively intact specimens. This is a baby mammoth from 40,000 years back that was found frozen in the ice of northern Russia. One side effect of the climate change is a lot of things that were basically embedded in ice for thousands of years are now turning up, right? As this ice is melting back, it's starting to reveal things. And so now if this is, these guys should be following protocol and should have masks over their faces because you don't want to be breathing your bacteria and germs all over these guys, but be that as it may, it gives you a chance to see, they can actually dissect some of these and find inside, what the thing was eating, right? What parasites it had. So this is not replaced by minerals. This is the actual organism. So sometimes you come up with really cool stuff like that. So as I said, not all fossils are the actual remnants, right? So they could be a cast. Sometimes they die in the mud and decompose, but that shape it held in the mud was left behind, and minerals infiltrated and fill it up. And so you wind up with a mold, a shape of a living thing. It's not the living thing itself. Imprints, like the footprints, or things like burrows, or coprolites, which are fossilized poop. And you might say, what would you learn from fossilized poop? There are things you can determine, like the form of the latter part of the digestive tract of the critter, what it ate, things like that. So I won't bore you with those stories. Onward. So paleontologists, paleo means old. Paleontologists are people who study fossils. So again, some are the actual organisms, some are things that are like imprints or the poop they left behind. Now, the fossil record is the ordered sequence of fossils as they appear in those rock layers. And that gives you a way of observing geologic time, because the newer stuff is on top of the older stuff, right? So if you think if your house gets cluttered, right? The clothes on the bottom of the pile went there before the clothes that are on the top of the pile, right? So because younger strata, younger layers are on top of older layers, you can determine the relative ages of fossils by where you find them. Also, some people will do is if they find something, they try and see what other organisms were found there. Like there's some things that are better known, like certain beetles or snails or whatever. And if you're trying to figure out how old the thing you have is, oh, there's these beetles here. And we know these beetles lived from, this many million years ago to that many million years ago. So it's not even just the layer of the rock, you can also use surrounding things to help you out with that. Now, Darwin acknowledged the fossil record is not complete, right? First, many of the things that lived on Earth didn't live in places that favored fossilization. Fossilization works best when you have fine grained soil or something like amber, or something can, if you think about fine grained soil, go to like the silty areas, or again, I think of low tide on the beach, but if you go to areas that have this really fine grained sand or fine grained soil called silt, if something dies and falls into that and gets covered up before anything can come and eat it, there's not a lot of oxygen in there. And so these fine grained, low oxygen environments pretty much don't favor something coming along and eating it, but do favor keeping it intact. Right? The silt, gets layers and layers and layers, and now you've got something embedded in this fine grained material. So not everything lived in a place that favored that. And then the stuff that did live in places that favored that were in rocks that later got destroyed, right? Earth moves, you get erosion from rain and rivers and things like that. So, not many things fossilized. Out of those, many were, destroyed just by the behavior of the Earth. So of the few that survived that, not all of them are accessible to paleontologists. So it's a sort of thing where it's fortunate that we find them. And sometimes we find awesome stuff that we didn't think we were ever going to find. But it's definitely not a complete record. You don't have a full lineage, like a pedigree of organisms from origin to modern day or to point of extinction.
This footage.
Okay, we're skipping over the Grand Canyon, but yeah, so you can see here, this is a good example. You see that little river down there, where over many, 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 many years, water moving through this piece of land cut further and further and further down into the land itself. And if you get up close, you can actually see layers, the actual strata. So it's kind of cool that you can actually see good examples of this. Now, you might say, all right, so how do we figure out things are related? Similarity that comes from common ancestry, right, shared ancestors, is known as homology. Again, that prefix homo meaning same. So if you are similar looking to another organism because you are closely related, we refer to this as homology. So characteristics present in an ancestral organism, again, tweaks over time by natural selection, right? Its descendants are facing different conditions because the world is changing. So as a result, you can have things that are related that have characteristics that are similar but function really differently. And a wonderful example of this is the anatomical similarities among vertebrate forelimbs as evidence of common ancestry. So if we go to this next illustration, all right, you see the forelimb of a human, a cat, a whale, and a bat. Now, if you look at these, you can see the bone that's closest to the torso, the uppermost bone in these pictures, right? We call it the humerus, right? The humerus of a human and a cat are fairly similar, but you look at the whale, and this is not really a limb for locomotion at this, at that point, an organism that's evolved for swimming. The tail does most of the propulsion. The fins are basically there mostly for steering. So you see that very short, very stocky humerus. Or you look at a bat where, okay, this form is kind of similar, but it's very thin and delicate. Why? Because if you're flying, you want to limit your mass. You want to limit your weight, because the more weight you're carrying, the more energy it's going to take you to fly around. So you see these long, delicate bones. And so you can work your way down each of these forelimbs, and you can see, oh yeah, there's ways where these limbs are similar here and similar there. But you get down to the digits, right, and the human phalanges that we're all familiar with. And if you look at the cat is sort of walking at the base of its toes, and so you can see the way that the heel is a little bit further off the ground. If you look at the yellow bones, that's the wrist, and that is comparatively the wrist on the cat. So a cat's only walking on the forepart of its foot. And it has things obviously like claws for adapted for grabbing prey. But you get to the whale and the bat, and you have elongated, flattened surfaces for steering in the water or actually carrying out flight, in the air. So you could tell they're all related. They all came from common mammalian ancestors, but they've been modified from the original ancestors. So when these organisms started living in these different places, their bodies eventually changed. Mutations that were beneficial were retained, and those variations get acted upon by natural selection. So it isn't something where they can say, oh, I could really use a really long forelimb here. It doesn't work like that. It's changes in the genes and also tweaks that happen in development. So it's not intentional, but over time the environment sort of edits the form of living things. So now this is how it was done for many, many years. It's just straight up anatomical comparison. But now, again, we have molecular biology. We can actually, trace the evolutionary history of things by DNA inherited in the ancestral species. And sometimes we can actually find stable DNA in preserved organisms. I knew a guy from New Zealand who studied the ancestry of ancient birds. And it turns out that there are these tubes where volcanic lava flowed and left these little cave-like spaces. And sometimes these birds fell down in there. These are terrestrial birds that didn't fly. And they sort of stumbled around and unfortunately eventually died, but there was nothing down there to eat them. And so they just lay there undisturbed in this, nice, cool, stable environment. And he was actually able to extract identifiable lengths of DNA out of some of those specimens. So sometimes we, like that little frozen mammoth back there, I'm sure they were able to find DNA that they could take out and analyze, right? So see the differences between that little guy who was frozen in the ice and the elephants that we have around today. So molecular biology has given us another level of looking at homologies, right? Instead of homologies of limbs, homologies of genetic material. So if you have two species with homologous genes with sequences that match closely, then they probably were inherited from a fairly recent common ancestor, that they diverged pretty short time ago from each other. On the other hand, the more sequence differences between species, well, then they diverged from each other much longer time ago, right? The more distant is the last ancestor that they both shared. So molecular biology, again, more evidence that all life forms are related, right? All living things use that same genetic language of DNA and RNA. The genetic code, how RNA triplets are translated into amino acids, is essentially universal. So organisms as dissimilar as humans and bacteria actually share some homologous genes inherited from a really distant common ancestor. So some of these things we share all the way back to our ancestry. Now, other evidence of evolution. So we have fossil record homologies. And within homologies, there's something called vestigial structures. These are remnants of features that were once functional structures, but from disuse, not being needed anymore, any mutation that reduced it in size or functionality wasn't damaging, right? If you think of cave fishes, right, these are fish that evolved from regular out in the world fish, but as they lived longer and longer in the cave environment, any mutation that caused damage or reduction of the function of eyes didn't hurt, right? Didn't do any damage. It's living in the dark. And so over time, the eyes, any mutation that had negative effect on the eyes tends to lead to the eyes being more and more reduced to the point where you just see little remnants left, but they're not functioning structures. If you look at fossils of ancient whales, you can actually find pelvis and hind leg bones from the ancestors that were originally on the land. And as they transition to a fully marine organism, you can see how the leg limbs changed. And just in your own body, if you pull up a sleeve and blow on your skin or stick your arm in a cold place or something, you get what? Goosebumps. They call it goosebumps because it looks like when you pluck a bird's feathers, you get those little bumps. those little bumps are caused by erector pili muscles. You have little muscles whose job it is to lift up the skin to make body hair stand up. Now, obviously, we do not have much in the way of body hair to stand up, either to signal to another individual or to try and stay warm or whatever, but those muscles are still there. And so you can, stick your arm in a cold place and you can watch, the muscles, lift up those tiny little hairs and you can still see them there, but again, not really functional. It's essentially a vestigial structure in us now. So homology can explain things that otherwise would be confusing. Good example, vertebrate embryos. At some point in their development, every vertebrate embryo, mammal, reptile, fish, whatever, has a tail posterior to the anus and structures called pharyngeal slits or pharyngeal pouches that as that embryo develops into an individual, if it's a fish, those throat pouches develop into gills. Now, looking around, you don't really see people with gills. So what happened to them in us? Well, they become parts of the ears and throat in humans. In fact, the little tube, if you've ever had to yawn or chew gum going up in an airplane or even up in an elevator, what happens is you're making the pressure within your head and the outside world even. And it's through a little tube. It's our last little remnant of a gill slit, and that's the eustachian tube in your ear. So let's see an example here. All right, so this is comparative embryology. So looking at the chicken embryo on the left and the human embryo on the right. I'll get to a closer picture in a second, but you can see the pharyngeal pouches. You can see the postanal tail, even though if you look at a chicken, you don't see a tail. And if you look at a human, you don't see a tail. So looking at it a little more closely, it's probably better if you look at the picture in your book. But there's these little pouches, these little slits, that are remnants of our filter-feeding ancestors from way back when, but they still show up in the embryonic form. And in some, like a chicken, you really, you could trace them into certain elements of the jaw and the mouth, but they're no longer there as throat pouches or throat slits. And again, that tail disappears during development. So again, pharyngeal pouches and then that little postanal tail, which during development, is degraded and lost. Now, sometimes people have like a little tag of flesh or something. That's just a little developmental oops. It does not mean that they are reverting to some other organism or something. So he visualized the history of life, because now life has a history, as a tree. And we often show these patterns with what's called an evolutionary tree. Now, it used to be drawn, bottom to top, right? At the bottom were the ancestral organisms, and you go out to the tips of the branches, and those are the modern forms. But they often now turn the trees sideways, and you read them from left to right, like reading English in a book. Homologous structures, both anatomical and molecular, can then be mapped, right, to determine how long ago things diverged, either by their anatomy or how long ago they diverged by changes in their genetic material. So they can then work out the branching sequence of an evolutionary tree. Now keep in mind, any evolutionary tree is a hypothesis, right? It's an answer to the question, how are these things related? So the one you see today, at a granular level, at a fine level, not the big scale of like fish versus amphibians, but if you're trying to determine how certain mice evolved, you might have some tweaking over time, right? So every time you see a tree, it's a hypothesis. Now, some are backed up very, very well, that at the higher levels of classification, right? So if you're getting down to, family and order and some of these smaller levels, there's still a bit of, discussion left. So the higher levels we kind of sorted out. But these are all hypotheses. And we support these hypotheses, these evolutionary trees, with data. And this is where I used to watch the taxonomists just about get into break a beer bottle fights in the bar. There was a bar near Yukon where these guys hung out. And just arguing what was an important feature to determine whether you lumped these tapeworms together or separated them. Yeah, these are tapeworm guys. So they are hypotheses, and scientists will argue about them, but again, ultimately it's a subjective human construct. Now, here's an evolutionary tree of tetrapods. Tetra meaning four, pod referring to foot. These are four-legged animals. And if you start from the left, right, you see this series of branches. Each time you have a branch point, each time you go from a line to two lines, that represents the common ancestor of the lineages that then extend from that point, right? So that uppermost one, you have a branch point where one branch goes to lungfishes, the other branch goes to everything that's going to have four legs. So every time you see that, that's the point at which they last shared, lungfishes and tetrapods last shared common ancestry. Right, so then we go down to the next branch, right, and we see this line diverge into two lines. Well, the first branch was things that have four limbs, right? If you look at the guys on the right, you've got amphibians, mammals, lizards, crocodiles, etc. All right, so that first branch is everybody right of there has tetrapod limbs. If you go to the next branch, the amphibians are separated out from everything else. Everything else has something called an amnion. It's containing an embryo within a fluid where it's supported and kept wet and all that good stuff. All right, so everything to the bottom branch of number two has an amnion. Number three, you have another branch, right? So you have things that bear the embryo and its amnion within the body during development. Everything to the lower branch on down bear eggs, right? The amnion is contained within an egg. And so from there, you branch them off in things like feathers, right? Separating birds from crocodiles and also dinosaurs. Because they're extinct, they're not in this illustration, but believe it or not, crocodiles and dinosaurs and birds are more closely related than crocodiles and snakes and crocodiles and lizards. So again, there's neat stuff out there. So if you look far to the right, you'll see a series of columns, right? And it's just showing you the progressive, from larger box to smaller box, right? So the big box being tetrapods. Within tetrapods, you have amniotes. Within amniotes, you have reptiles. Within reptiles, you have birds. And yes, birds are technically reptiles. Why? Well, that branching point I show, right? So if you look at branching point 4, that's a common ancestor. Everything past that point is a related group. Actually, probably easier to say 4, the point of the amnion, leading to shelled organisms. So think of 3. You see that line extending from 3. Everything past that point, past that line that goes down and to the right, is related. They're all in the same group. You can't just pluck something out and say, okay, this has different ancestry. It doesn't. They all share ancestry. They're all related. And that's why birds are now classified as reptiles. Okay, so natural selection affects individual organisms, but individual organisms do not evolve, right? You as a person cannot evolve. You as part of a population, your population can evolve, but you as an individual do not. So a population is a group of the same type of organism living in the same, general area at the same general period of time. So you and a person who lived back in 1805 would not be a member of the same population, right? You're the same species. You might be, all both living in a haven, but it has to be a cohort. It has to be a contemporary, a group that lives at the same time. So a population, the group of organisms, that's what evolves over time. As adaptive traits become more common in the group, and other traits change or even disappear. So when we talk about evolution, we're talking about generation to generation changes in populations. Now, because they hypothesized that species formed gradually over long periods of time, he knew he would not be able to study the evolution of new species by direct observation. He did conceive the notion that artificial selection, the selective breeding of domestic plants and animals to promote traits that we desire in those organisms, right? So it's not determined by nature, it's determined by humans choosing, which organisms breed and which offspring are bred again. So this business of artificial selection was a great way to look at and understand evolutionary change, because we can actually see how people have changed things over time. Now, I just realized there's a typo. Darwin realized that Natural selection has two essential components. Actually, both types of selection do, but good mercy, I never noticed that before. So Darwin learned that natural selection, or artificial, since we're the selecting value, has two essential components. One, variation among individuals, and two, heritability, the transmission of a trait from parent to offspring. So he knew that individuals in natural populations have small but measurable differences. But he didn't know what forces in nature were determining which individuals were going to be the breeding stock for the next generation, which individuals were going to leave more offspring in the next generation. So they show you a bunch of lady beetles here. And this is a single population, right? They were all collected from the same place at the same time. And you can see some have no spots, some have an orange or yellow background, some have a black background, right? There's all this variation in this population. So if there was something in the environment that favored either the light background or the dark background, or fewer spots over more spots, if there's something in the environment that favored that, think of something that's eating bugs for a living. If you're more easily visible, you're more likely to get eaten, right? So if there's some feature in the environment that favors some of those guys more likely to survive and leave fertile offspring, then those are the guys who are winning at natural selection. Now, there's a guy named Thomas Malthus. You might hear the term Malthusian or Malthusian. He contended that human disease, famine, and war, like what we're going through right now, were the consequences of human populations increasing faster than resources. So Darwin took some of that away from the kind of not too good for human society elements that he had in there, but he took the idea of populations of plants of animals increasing and what limits them. And he said, we reasoned that, okay, the resources of any given environment are limited. You don't have an infinite amount of space, an infinite amount of food, etc. So the resources of any environment are limited. The production of more individuals than the environment can support leads to a struggle, right? If everybody is eating the same stuff, not everyone's going to get enough to eat. So the production of more individuals leads to a struggle for existence, and only some offspring survive in each generation. And just to give you an example here, so looking at this little mollusk here, if you look at that white band down at the very bottom of the picture, you can see the tiny little dots. Each one of those dots is an egg. So you're probably looking at thousands of eggs being laid in this ribbon all together when she's done. If each one of those grew up into its own little sea slug, then you would have sea slugs filling up the place, right? So more offspring are produced than are going to survive, right? Some of them are going to get eaten. Some of them the eggs may wind up in a place where they can't develop properly, right? You make more offspring than are going to survive to adulthood and reproducing themselves. So in the process of natural selection, individuals whose traits better enable them to obtain food, escape predators, or tolerate physical conditions, these are the ones that survive and reproduce more successfully, right? And they now pass their heritable traits onto their offspring. And Darwin reasoned if artificial selection can bring about pretty significant change in a relatively short period of time, then natural selection could modify species considerably over hundreds or thousands of generations. Now, we have actually managed to document evolutionary change in many, many studies. The one about insect species is kind of unsettling because this is something that, was in the last century. And so I'm just going to pop to it. Basically, one thing it says here, pesticides cause pesticide resistance in insects. It doesn't cause resistance. It's like the filter, right? You have all these organisms and you put the filter in of able to survive a pesticide. And if there's variation in that population, enough are different enough that they survive, right? So the filter only lets through those individuals that are resistant to the pesticide. They didn't make it for the occasion, but they had that variation already there because of some mutation back along the way. So the pesticide is basically just a filter right? It's a selective element that selects everybody who can't survive. It selects against anybody who can't survive pesticide. So the pesticides don't cause resistance, but they promote those alleles in a population, right? So they might have been 1 out of 100 in the original population. Well, when they become 1 out of 2 in the survivors, right, then, okay, this guy is definitely going to be more successful at leaving offspring because doesn't have to share, got more real estate, more mates available, blah, blah, blah. Now, natural selection, therefore, can only amplify or diminish things that are heritable. Right? During its lifetime, an organism can acquire characteristics that help it survive. But if it's acquired during the lifetime, now again, sort of set aside epigenetics for now, but if it acquires characteristics that helps it survive, then that's something that doesn't get passed on, right? So pierced ears or big muscles or whatever, those don't get passed on to offspring. So something to keep in mind also, natural selection is not goal-directed. It doesn't lead to perfectly adaptive organisms. A trait that is great in one situation may be useless or even harmful in another. The actual genes associated with DDT resistance, it actually was linked to things that made, before DDT, made mosquitoes with those genes a little less successful. They kind of sucked a little bit in some features of life. But once you wiped out everyone who, didn't have resistance, then suddenly that was the good set of genes to have. So when situations change, it often leads to, what was good one day is not so good the next, or vice versa. And the next slide just, shows you an example of pesticide resistance, right? So the initial population is up at the top. And you got that one, a couple of guys there that have an allele conferring resistance to pesticide. The only ones that survive have that allele. So now these are the ones that are leaving, pretty much all the next generation, right? So the more individuals the fertile individuals you put in the next generation, the more successful you are in the Darwinian sense. So things like this are not a good long-term solution. Now, one thing Darwin didn't know is that how the variations that are this raw material for natural selection arise. So he didn't know didn't know about mutation. He didn't even know specifically how those variations got passed from parent to offspring, the elements of heredity, even though Gregor Mendel had already answered these questions. But back in the time of not everything being connected by the internet or other means of communication, this guy had already worked this stuff out, but Darwin never got a chance to learn about it. Now, individual variation occurs in all species, and most populations have obvious physical differences, right? Humans are great for this, right? We have a huge number of differences, among individuals. So most populations have this phenotypic variation that really, the ones that are obvious, and there's ones that you can only see at the molecular level, right? So some things are easily visible. Some things you'd have to drill all the way down to look at, how proteins function or something like that. Also, again, not all variation in a population is heritable. Only the genetic component of variation is relevant to natural selection. And here's another example of variation in a population. Right? You have all these snails, and there may be differences in the patterns, but if somebody got their shell cracked by something, that wouldn't be passed on. But the pattern of colors that it got from its genes, yes. So as I've been saying, mutation is the ultimate source of genetic variation that serves as raw material for evolution. New alleles are going to originate by mutation. This is a change in the nucleotide sequence of DNA, right? You did this back in Lab 9. In multicellular organisms, only mutations in cells that produce the gametes can get passed on to offspring, right? So if you have a mutation in a skin cell, that's not going to get passed on to the offspring. So it's got to be mutations in, in this case, in cells in the ovaries and testes for our own species. Only mutations in cells that make gametes can get passed on to offspring and therefore affect a population's genetic variability. But it can be a small change, right? The change is as small as that one single nucleotide in a protein coding gene, leading to, the misshapen hemoglobin and leading to sickle cell disease. Now, if you remember back, right, protein shape and protein function were closely associated. So mutation that affects a protein's function will probably be harmful. On rare occasions, a mutated allele may improve the adaptation of an individual to its environment and therefore enhance its reproductive success. I remember a professor who said, mutation is kind of like walking up to a car and putting a bullet through the hood. It may break something important, it may go completely through and doesn't change anything, or it may knock something into a better position. Now, the odds are it's either going to break something or pass all the way through more than make something work better. So mutations that affect the function of a protein tend to be harmful, but now and then you get mutations that wind up being beneficial. So we see this effect more when the environment is changing rapidly enough and in such a way that a mutation that was once a disadvantage becomes an advantage. And I'm setting up, anyway, chromosomal mutations that delete, disrupt, or rearrange many loci at once are almost certain to be harmful. But duplication of a gene or small pieces of DNA through errors in meiosis can provide an important source of genetic variation and eventually lead to new genes with novel functions. There's a whole business of genes that control development. And it's thought that pieces getting copied, more copies of a gene winding up in organisms, leading to repetition of sections. So if you think of an insect, right, has those three main sections, each of which has a pair of walking legs. It's thought that there might have been, say, 4 legs, and another pair, goes in there because there's a duplication of a gene. There's different examples out there. Since we didn't get into gene control, I won't get into that here. But sometimes the duplication can lead to something functional. In prokaryotes, mutations can quickly generate genetic variation because what? Bacteria are haploid. They just have a single allele for each gene. And what do? They reproduce asexually, right? They just divide. So one bacterium that splits in two and those offspring split in two and those offspring split in two, on and on and on, then what? That mutation, that change in gene may suddenly create a lot of variation in a population in a fairly short time. So sometimes an allele can have an effect immediately. Now, multicellular things like plants and animals, they average about 1 mutation in every 100,000 genes per generation. Now, plants and animals have a long time span between generations, right? It's not minutes to hours, it's often years, often many years. So animal plants also have diploid genomes that prevent most mutations from significantly affecting genetic variation from one generation to the next. Because if you have a homologous, allele that is, that might mask, be dominant over that novel mutation, then it might get masked, and you won't see it pop up. So it looks a bit slower in things like plants and animals. Okay, so in organisms that reproduce sexually, most genetic variation in the population results from that unique combination of alleles that every individual inherits from its parents' gametes, right? Because think about that. Remember, now we're going to go back to meiosis here. Fresh assortments of the alleles existing in these parents arise every generation from the 3 random components of sexual reproduction. You're going to see this on the test. 1, independent orientation of homologous chromosomes at metaphase 1 of meiosis. 2, crossing over. And 3, random fertilization. So because of these 3 components, you're really shuffling the genetic deck a lot in sexually reproducing organisms. So, let me just skip over
In sexually reproducing organisms, 3 processes lead to most genetic variation. independent orientation of chromosomes in meiosis, crossing over of chromosomes in meiosis, and random fertilization. Each pair of homologous chromosomes consists of one chromosome inherited from the father and one from the mother. Here we have color-coded them blue and red. Each pair of chromosomes lines up independently of the other pairs in metaphase 1 of meiosis. Here you see one of the possible arrangements and outcomes. There are two different ways that each chromosome pair can line up. That means that in the organism shown here, with the diploid number of 4, independent orientation of chromosomes at metaphase 1 can produce gametes with 4 different combinations of maternal and paternal chromosomes. In a human being, with 46 chromosomes, more than 8 million combinations are possible. Now let's look at how crossing over creates even more genetic variability. During prophase 1 of meiosis, homologous chromosomes pair up very closely, and corresponding parts of two non-sister chromatids may trade places. This process of crossing over creates variation by producing chromosomes that combine the genes inherited from two parents. Here the process produced a total of 4 genetically different gametes. There are many ways crossing over can occur. In humans, crossover events happen an average of 2 or 3 times per chromosome pair, greatly increasing the variation among eggs and sperm. Note that crossing over produces some parental gametes with chromosomes like those of the parents, and some recombinant gametes with a mixture of genes from both sets of chromosomes. Independent orientation and crossing over occur simultaneously during meiosis, multiplying the number of genetic variations among gametes. Because each pair of chromosomes lines up independently, and crossovers can occur almost anywhere along each pair of chromosomes, it is possible for a human being to produce an almost infinite variety of gametes. A sperm fertilizes an egg, producing a zygote. The random nature of fertilization adds to the variation arising from meiosis. Each parent is capable of producing a huge variety of genetically different gametes. The number of possible combinations among their offspring is staggering. Theoretically, one human couple is capable of conceiving a number of genetically different offspring that is far greater than the number of humans who have ever lived.
Okay. Skipping over to here. Now, as I said before, individuals don't evolve, but populations do. So, the evolutionary impact of natural selection is apparent only in the changes in a population of organisms over time. So again, a population is a group of individuals of the same species that live in the same area and interbreed. Different populations of the same species may be geographically isolated from each other to such a point that exchange of genetic material never or only rarely occurs. And a great example of this is populations confined to different lakes. So we have this photograph from Alaska where you have these lakes, they're very close to each other, but if you're talking about something like fish that are not going to get out and scamper from place to place, there aren't the walking catfish, we're not talking about that, just your regular fish. Well, if the fish aren't going to be moving from pond to pond, then they're effectively isolated. And those populations are not going to be interbreeding with each other. At the population level, biologists focus on the gene pool. That's all the copies of every type of allele at every locus in all members of that population. So for many loci, you've got 2 or more alleles in the gene pool. Now the next few slides are going to have a blue line through them. Those are the ones that we're going to skip over. This is getting into something called the Hardy-Weinberg formula. So I am sparing that from you folks. So the slides with the line, you don't have to worry about that for the exam. So 1, 2, 3, 4, 5, 6. Okay, so don't worry about these guys. Now, the next slide I left in here because the one important thing to realize is a non-evolving population is in genetic equilibrium, which means the gene pool remains constant. The frequency of alleles doesn't change. Now, in the real world, That almost never happens because what? The world keeps changing. So this whole business of Hardy-Weinberg equilibrium is more of a concept than something you really see very much. Now, the second paragraph is the more important one here. Because a generation to generation change in allele frequencies of a population is evolution on the smallest scale, we sometimes call that microevolution. So do know the term microevolution. So, what changes the gene pool? Well, the three main causes of evolutionary change, again, you see a list, you know you're going to see this on the test. Three main causes of evolutionary change are natural selection, genetic drift, and gene flow. Now, natural selection is the most important because it's the only one of the three that promotes adaptation. Right? So keep in mind, you have three things that can cause the change of frequencies of alleles, but only one is going to lead to adaptation, and that's natural selection. So let's start looking at the other two since we've done a bit on natural selection already. Genetic drift. Genetic drift, when you hear genetic drift, think random. So this is a change in the gene pool of a small population due to chance, right? Stuff happens. If you're the flower in the field that gets stepped on by the person walking through it, it probably had nothing to do with your genes. You were just in the wrong place at the wrong time. Now, the bottleneck effect is an example of genetic drift. This results from a drastic reduction in population size that, one, decreases the overall genetic variation in a population because at least some alleles get lost from the gene pool, and two, results in a loss of individual variation and therefore adaptability. So I'm going to show you these flowers. So again, don't get too hung up in the numbers. Just I'm going to show you that the numbers change, but don't, again, you're not going to worry about the calculations or anything like that. So say we got a bunch of flowers here, and so we've got a combination of purebred red, heterozygous red, and purebred white, right? So those are your three possibilities. And the whole business of allele frequency is you count up the number of alleles. And you might say, well, there's so many in the, in these homozygous dominant, so many in the homozygous recessive, right? But you also have to count the hets. So it's the number of each of those plus, the numbers that you see in the het. So bottom line, if you count up all the letters here, you've got roughly 7 of the dominant alleles to 3 of the recessives, right? Basically, P plus Q equals 1. All right, so think of it like percentage. This might be the easier way to think of it in a generic sense, right? So 70% of P, 30% of Q, add those up, it's 100%. So something happens, tree falls, a rock, gets dropped somewhere. Something happens that wipes out most of the plants so that only 5 survive and leave offspring, right? So you look at the second generation, and now we see the P and Q ratio is now 50-50, right? Because you lost a bunch of the reds, and again, this was random, nothing chose it, but just because whatever happened to these flowers, only 5 left offspring. Well, now you're going to have from these parents, right? They're all red in color, but most of these guys are carriers. So next generation, you're seeing a slightly higher ratio of the white, right? So instead of 70-30, it goes to 50-50. All right. Other things happen. Right? It's only 2 of 10 plants in this crowd leave offspring. Well, if the only 2 that survive are those 2 that are circled, that's what they indicate the survivors by the circled ones. Well, if the only survivors are homozygous dominant, then what? Your third generation goes to 100% on that dominant allele and 0 on the recessive. So something like genetic drift can really cause drastic change in a fairly short period of time. And the reason why they call the bottleneck effect what they do is think of the original population as being a whole bunch of alleles in a bottle. If you take that bottle and just shake a few into a jar, odds are you're probably not going to shake an even proportion of all those alleles into that jar. In fact, some colors you can see don't make it in there at all. So as a result, your surviving population, you've lost all the oranges, right? You only have 1 green and a few of the purples. So that's your new gene pool. So you go from all that variation in the original population, a bottleneck event, and then your surviving population has much less diversity. So here's a video.
Variation is found in human population.
Oh, sorry. Back again.
Variation is found in human populations and in other populations, such as this population of island beetles. Color in these beetles is determined by different versions of a gene, different alleles. Green is the dominant trait, so green beetles have two green alleles, or a green allele and a brown allele. Brown beetles have two brown alleles. By counting the brown alleles and green alleles, we can calculate that this population has 30% brown alleles and 70% green alleles. The frequency of brown alleles is greater than the frequency of brown beetles because some green beetles have a brown allele. Allele frequencies change as populations evolve. What causes populations to evolve?
So watch that bar on the left.
The island is suffering from a drought, and in this dry environment, brown beetles are better camouflaged than green beetles. Birds are more likely to see and eat green beetles. Over time, brown beetles survive and reproduce more than green beetles. This is an example of natural selection. Individuals with inherited traits better suited to their environment survive and produce more offspring than other individuals. Here, natural selection causes the frequency of the brown allele to increase. Through natural selection, the population has become better adapted to its new environment. Genetic drift occurs when populations evolve due to chance events. Let's see how genetic drift affects this small population of beetles. Chance constantly affects which beetles actually reproduce and which of their alleles they pass on. In a small population, chance events produce large fluctuations in allele frequencies from one generation to the next. Here, the frequency of the brown allele has decreased to 0, and the population is left with only green beetles. Genetic drift also occurs in large populations. But in a large population, genetic drift has less effect because it causes much smaller fluctuations in allele frequencies. Other random events can also lead to genetic drift. By chance, this volcano kills more brown beetles than green beetles, causing the frequency of the green allele to increase. Gene flow describes evolution due to individuals moving into or out of a population. When green and brown beetles migrate into the all-green population, the frequency of the brown allele increases. Natural selection, genetic drift, and gene flow are the major mechanisms responsible for the evolution of all populations. Together, they shape populations of beetles as well as humans.
Okay. Now, cheetahs, which are a cat most of us are familiar with, appear to have experienced at least two genetic bottlenecks. One during the last ice age about 10,000 years ago, and a second one in the 1800s when farmers hunted the animals almost to extinction. So, because you've had two bottleneck events, you've really lost a lot of the variation that was in the original population. With so little variation, cheetahs today have a reduced capacity to adapt to environmental changes. One of the greatest fears of conservationists would be something like a disease, that if there was no variation, possibly providing an element of resistance, then they're all at threat. Right? There was research done where they took skin grafts. You know that organ transplants require pretty close matching, of genetic markers, and so do skin transplants. Well, they're able to take a skin graft off of one cheetah on one side of the continent of the African continent, and grab a cheat on the other side of the continent and put on that skin graft, and it was not rejected, which means that they're all closely related. So we have to be careful with these guys now. Now, the founder effect is another random element. This is likely when few individuals colonize an isolated habitat. So this would be genetic drift in a new colony, right? So something causes a few individuals to get isolated from the rest of the population out there. It also explains the relatively high frequency of certain inherited disorders in small human populations. If you think of folks like the Amish, where they may not live geographically very far from other people, but culturally, they tend to marry within their own population among people of similar faith. So, in effect, they are isolated, right? We've taken this population and dropped them somewhere, and there's no gene flow. Right? There's no genes coming in or out. Similarly, folks like the Hasidic Jews, right? They have very strict, rules against marrying out of the faith. And so you could be in the middle of New York City, and where you have a large population of Hasidim, they may be in the middle of the largest city in the continent, but they're in effect, genetically isolated because there's no gene flow between their group and the rest of the surrounding human population. Now, gene flow, this is another source of evolutionary change. It's genetic exchange with another population. Now, this can either result in gaining alleles or losing alleles. And it tends to reduce genetic differences between populations, right? So if they're isolated and there's no gene flow, right, they tend to become different. But if everyone's sharing genes back and forth, it tends to average out, right? It tends to reduce the differences between populations. And some types of gene flow are pretty much baked in the cake. This is a tree, and all those little green dots are pollen grains. Pollen grains that are borne by the wind. If you have something that's pollinated, where the eggs are being fertilized by wind, well, then wind can carry those pollen grains all over the place, right? So one form of gene flow is even things like this. The way in which an organism reproduces may be contributing, maybe an element of gene flow itself. Now, of those three, again, only natural selection consistently leads to adaptive evolution. Right? This is evolution resulting in organisms better suited to the environment. So, give you an example here. Structure and function. Back to structure and function. There's a bird called the blue-footed booby that's very well suited to where it lives in the Galapagos Islands. If you look at the bird, it's very streamlined. Right? The body and the bill are tapered at both ends. At the end of one end of the body, the tail is tapered. The front end, the head and the beak are tapered. So when it dives, when it's flying and then dives into the water to catch fish, right, it's more tapered, so it minimizes friction, right? It's very efficiently goes through the water. The big tail can be flared out to act as a brake, right? These things are flying above the water, then they fold everything and zoom down, and then they can flare out the tail to serve as a brake so they don't, hit the water too hard. And they have a neat feature. Female boobies like the males with the brightest blue feet. And here's a handsome fellow right here. So they have this very elaborate dance in which the male shows his beautiful blue feet to the lady. And that is something which makes the girls choose these guys over the others. We'll be into that in the next chapter. So the commonly used phrase survival of the fittest, which you tend to hear, is misleading because we take it to mean like it's head-to-head competition between individuals. It isn't really quite that. Reproductive success, that's the key to evolutionary success. That's generally a little more subtle and passive. It isn't like you see two snakes go out and who can lay more eggs than the other? No, it's not quite like that. Relative fitness is the contribution any individual makes to the gene pool of the next generation relative to the contributions of other individuals in that population, right? So he or she that leaves the most fertile offspring wins. That's what relative fitness is. Now, the process of science example they have here. Did natural selection shape the beaks of Darwin's finches? So along with interesting lizards and tortoises on those islands are the very famous Darwinian finches. That finches on these different islands evolved into somewhat different forms. Again, we now realize they've adapted to the food sources on each of these islands. So biologists hypothesize that Darwin's finches evolved from a small population of ancestral birds that colonized one of the islands. Natural selection shaped the beaks of finches to make use of diverse foods in this new environment. And for 40 years as of this writing, I think getting closer to 50 now, these two ecologists, Peter and Rosemary Grant, and their graduate students since then, have studied Darwin's finches on Daphne. It's a very, very tiny Galapagos island. And because it's so small, it was fairly easy to collect comprehensive data on the organisms that live there. Well, the climate of Daphne alternates between wet and dry seasons. So during the wet season, lots of rain, the plants bloom, there's lots of seeds to eat. When it gets to the dry season, it's really dry, and food becomes very scarce. So they wanted to test the hypothesis that the beak of the medium ground finch changed in response to changes in the available food. And the results they observed was the proportion of birds with larger beaks increased in the population during this intensive period of natural selection. There was a severe drought, so it wasn't just normal wet dry. There was an actual drought. It was prolonged dry weather, and that led to the plants making tougher seeds. So here's a picture of the Grants. Here's the bird in question. So if you look at this medium ground finch, if you look to the little inset on the left, you see that white line with the arrow at each end. That's what they measured. So when they talk about beak depth, that's what they're talking about. So basically how broad the beak is vertically. And we go to the data here, and I think this is also in your book, so if this is hard to see on the screen, you can grab the book. If you look at the top of the top graph, you have rainfall on the y-axis and the years on the x-axis. So this is what you're looking at is rainfall during the wet season. So 1976 had a pretty good wet season, then it went dry, and then 77 was really dry. And that wasn't really fixed until 1978 when it went back to a normal dry season. So you had that chunk of food available for the 76 dry season and food available for the 77 dry season. So if you look now, follow these arrows down. So if we're looking at the food available for the 76 dry season, in other words, there had just been a lot of rain, so the food was blue line, abundant, there was lots of it, and orange line is relatively low, it's not too tough, right? So lots of food, easy to get to. Now you go to the 77 dry season, right? So after a very, very dry, quote, wet season, and now you look what happens. Seeds are in lower abundance, right? That blue line is lower, and they became much tougher because there's what? If there's not as many seeds, it's sort of, the seeds tended to develop stronger hulls, to be harder to open up and eat. So that's what happened with seed abundance and toughness. Now, let's see what happened to the birds. Well, 1976 before the drought is the bar on the left. 1978 after the drought is the bar on the right. And sure enough, if you look at the 76 bar, that beak depth, how broad that beak was much shorter than the population after the drought. And one problem was, one thing that also happened was because of the drought, they also lost a lot of birds. So this is a case where you're not really, you usually think, oh, what we're seeing over generation to generation. Well, here, selection basically knocked out the ones that could not consume those tougher seeds. So it wasn't even like who was successful, who survived. Only 15% of the original population of 1,200 or 180 birds survived. So the ones that survived showed that selective element, right? If you didn't have a broad beak, you couldn't crack the seeds, you didn't live, you didn't produce offspring. So it was a kind of a fascinating example where you can see selection on a very, very short-term scale. Now again, the variation was already in the population, just the ones that had the broader beaks were the ones that survived. Now, sexual selection is one type of natural selection in which individuals with certain traits are more likely than other individuals to obtain mates. So sometimes a feature that is beneficial in natural selection is going to be, so it may be something where a feature is good at getting mates, but it may not improve survival. Sexual dimorphism, di referring to two, morph referring to shape, is a distinction in appearance between males and females that, again, isn't directly associated with reproducing or surviving. So in a more common type of sexual selection, individuals of one sex, usually the females, are choosing and selecting their mates. So we look at the next slide. Right, you have these finches on the left where you can see a very distinct difference between the male and female. And on the right, you see a pair of ibex males that are competing for the mate. So the males fight to get see who wins, and eventually who wins generally gets selected by the female. Now, there's three general outcomes of natural selection. If you graph the coat color of a population of mice, you get a bell-shaped curve, right? So relatively light at one end, average in the middle, relatively dark at the other end. If natural selection favors certain fur color phenotypes over others, then the populations of mice will change over the generations. Now, there's three general outcomes that are possible if you place an environment that's changing and you have resulting changes in the population. One is directional selection, and that shifts the overall curve, right, the overall makeup of a population by selecting in favor of one extreme phenotype, right, so more to one side or the other. Second is disruptive selection, and that can lead to a balance between two or more contrasting phenotypes in a population. So instead of the whole curve moving to one side or the other, basically put a dent in the curve, right? You're going to have, two peaks in that curve. The third is stabilizing selection, and that favors intermediate types. So it's neither one extreme or the other, but something more in the middle. And stabilizing selection is the most common, and you see it in fairly stable environments. So you go to the next slide. Right, so up at the top you have the original population. Right, so frequency of individuals is the y-axis, the phenotypes are on the x-axis. So you see the fur color goes from light to dark. And anything where you have a gradation like that, you tend to see this sort of bell curve, right? So very light, very dark, and then most in the middle here. Now looking at the outcomes at the bottom. So if you look at the far left one, okay, this would be directional selection where, let's say, the environment got relatively dark. Say there was something where dark-colored plants became predominant, so light-colored mice were more likely to be turned into food. So you see that whole curve shift to the dark end. Right? That's directional selection, right? That curve is moving in one direction. In the middle, you have disruptive selection. So whatever selective elements are there, there's actually a favoring of the very light and the very dark, but not the ones in between. So now you see two peaks, one for the light-colored mice, one for the dark-colored mice. That's disruptive selection. And the last is stabilizing selection, where the peak doesn't move one way or the other, but it narrows. Right? You're selecting against both extremes and favoring what's in the middle. And that's what you see most commonly out in nature. And one example within our own species is head size of babies. If a baby has a too small head, there's probably a chance that there's issues with the functioning of the brain. But if the baby's head is too big, it may be lethal to the mother and also the infant at birth. So evolution has sort of favored, the in-between. So not too small, not too large, but just right. But in the modern era, now with the advent of all the caesarean sections out there, I wonder if you're going to see that stabilize, that the range of, that you're going to see broadening somewhat in countries where there's a lot of caesarean sections and remaining stable in countries where it's relatively uncommon. But that's research. I don't know if anyone's done that yet. All right, evolution connection. So again, antibiotic resistance. We all know this one, right? Antibiotics, anti against biotics life. So these are drugs that kill infectious microorganisms, not viruses, because viruses have none of the cellular elements that antibiotics are used to damage or impede. Well, back in the 1940s, penicillin was the first widely used antibiotic. By 1952, some bacteria had genetic traits that enabled them to resist the killing power of the drugs. In that same way that pesticides selected for resistant insects, antibiotics select for resistant bacteria. Right? The ones that survive have the resistance and are what? Have a higher relative fitness. They're leaving more offspring. So they've a series of diagrams here showing urgent, the deep red, and serious orange threats from antibiotic-resistant bacteria that affect humans. Right, so the orange or red refers to percentage of antibiotic-resistant cases. So the top one is a form of gonorrhea. The pathogen is Neisseria gonorrhea, and it's a generally sexually transmitted organism. Well, that one, there's a progressively larger group of antibiotic-resistant cases turning up. And if you go to the next one down, it's a form of pneumonia that, it's Streptococcus pneumoniae, which is typically associated with pneumonia, bloodstream infection, ear infection, even meningitis. And again, we're seeing pretty large proportion of these resistant forms developing in the population. A couple that are less of an issue so far are food poisoning caused by either Campylobacter, Salmonella, or Shigella. So fortunately, at least those are not totally taking over yet, but the fact that these are developing, because keep in mind, where do we often see these? In food preparation places. So if there are forms that are becoming resistant to antibiotics, then it's going to be harder to treat these cases when they do turn up. Almost done. So, evolution connection. Our enthusiasm for the curative powers of antibiotics has what? Encouraged the evolution of antibiotic-resistant bacteria. And something that's happened to make the things a little bit more challenging is livestock producers will add antibiotics to animal feed. Not because the animal is ill, but for some reason, antibiotics make the animal grow faster. And if you're in the business of growing as much cow as possible in a short period of time, then feeding the cow antibiotics, it's like, hey, I get more cow per season, and I make more money. Problem is now what are we doing? We're putting these antibiotics out in large volumes out in the world. So we're promoting, antibiotic-resistant bacteria by doing so, right? We're out there carrying out natural selection. Doctors may overprescribe antibiotics right now. During a flu epidemic, you have doctors who are being harassed into giving antibiotics. Any good doctor knows an antibiotic does not work against a virus, but either by being nagged by a patient or for whatever motivation, they may prescribe antibiotics that are not necessary or even useful. And third, people who do get antibiotics for the right reason, they may not take the full course. So you might have bacteria that maybe don't have perfect resistance to the drug, but are moderately resistant to the drug, right? You let the ones that you would have wiped out had you taken the whole series survive. And so again, you're promoting, individuals with any mutation that has resistance to bacteria. So those guys survive, those guys multiply. Over time, that gets fine-tuned by, these individuals going to other people, and maybe those people not taking the full dose. It promotes antibiotic resistance. So hospitals are sort of a brutal area to observe antibiotic resistance, right? Because these are areas where you have A, sick people, B, a lot of people getting medication. And MRSA, methicillin-resistant Staph aureus, is, as I'm sure all of you know by now, it can cause potentially fatal systemic infections, and you often see it in community settings. They're now realizing athletic facilities. So kids who are out playing soccer and football, that sometimes there's transmission of these in those settings as well as just hospitals, also in schools, even military barracks where you've got people crowded together and potential injuries making a nice environment available to the organisms. All right, that is it for the chapter. See you soon.